40 years. That's how long Lupron Depot has been on the market, treating prostate cancer, endometriosis, uterine fibroids, and precocious puberty. It's been injected into millions of patients worldwide. The patents have long expired, and yet today, right now, as you are watching this, there is no FDA approved generic versions of Lupron Depot in the United States. And why? That's what we are going to unpack today. And the answer goes much deeper than patents and legal strategy. It gets into very heart of pharmaceutical science, manufacturing complexity, and regulatory framework that was simply not built for products like this. Hi, I'm Raghuveer Singh Parmar, Director Scientific Affairs at Resolve Mass Laboratories Canada. Resolve Mass Laboratories stands at the intersection of analytical excellence and complex drug development, helping pharmaceutical teams overcome critical challenges in peptides, impurities, and regulatory pathways. Let me frame the question properly, because a lot of people misunderstand it. The active ingredient in Lupron Depot is leuprolide acetate, a synthetic nonapeptide GnRH agonist. Leuprolide itself is off patent, so you can buy leuprolide API. The molecule is well understood. So what's the problem? The problem is that Lupron Depot is not just leuprolide. It's leuprolide encapsulated inside a highly engineered microsphere delivery system, and that delivery system is arguably more complex than the drug itself. Generic drug development usually follows a simple playbook: same molecule, same dose, proven bioequivalence, and done. But Lupron Depot breaks every assumption behind this playbook. We need to understand the PLGA Microsphere platform. So let's get into the science. Lupron Depot uses PLGA, polylactide co-glycolide, a biodegradable polymer that has been in clinical use since the 1980s. On its surface, PLGA sounds elegant. You can encapsulate drug inside these microscopic polymer blends, inject them, and polymer slowly degrades in vivo, releasing the drug over weeks or months. One injection, sustained therapeutic levels, fewer hospital visits, better patient compliance, and it works beautifully. For the innovator, this has spent decades of perfecting it. But here is where it gets interesting. PLG is not a simple defined chemical entity. It's a random copolymer of lactic acid and glycolic acid, and its properties, its molecular weight, its lactide-glycolide ratio, its N-group chemistry, its blockiness are inherently variable. And each of those variables has a direct, measurable impact on how the drug is released from the microsphere. Think about this way. You could buy PLGA from 3 different suppliers, all with the same specification on their certificate of analysis. Same molecular weight, same 75-25 ratio, same N groups. Put them through the exact same manufacturing process, and you'd end up with microspheres that have different porosity, different glass transition temperatures, different drug distribution patterns, and different in vitro release profiles. The 3-phase release problem. Now let's talk about the drug release, because this is where the complexity becomes almost philosophical. Lupron Depot doesn't release leuprolide in a single clean wave. It follows characteristic triphasic pattern. First, there is an initial burst. Leuprolide sitting on or near the microsphere surface releases quickly in the first few days. This matters clinically. It's partly responsible for the initial testosterone flare that clinicians warn patients about. Then comes the diffusion phase. The drug slowly migrating through the polymer matrix over days to weeks, maintaining a therapeutic plasma concentration. And finally, the degradation phase. As the PLGA polymer hydrolyzes and its structure breaks down, the remaining drug is liberated. Now ask yourself, if you are a generic developer trying to match this profile, how do you even measure whether you have done it correctly? You can run in vitro release testing, yes, but the release behavior of a PLGA microsphere in a dissolution bath is not the same as in a human muscle. The correlation between in vitro results and in vivo pharmacokinetics, what we call IVIVC, is notoriously difficult to establish for long-acting parenterals. And if your bioequivalence study requires an in vivo data with monthly or 3-monthly endpoints, you are looking at clinical studies that run 6 to 12 months per formulation iteration at enormous cost. So let's talk about, let's move from chemistry to manufacturing, because the production of PLGA microsphere is frankly a craft as much as it is a science. The classical process is a water in oil in water double emulsion followed by solvent evaporation. You dissolve PLGA and leuprolide in an organic solvent, homogenize it with an aqueous phase to form a primary emulsion, that then disperse that into a larger aqueous phase with a stabilizer like PVA. As the solvent evaporates, the polymer hardens into microsphere with the drug trapped inside. Sounds straightforward enough, but every parameter in that process, for example, homogenization speed, temperature, phase ratio, stirring rate, solvent removal rate, drying conditions, all affects the final particle size distribution, the drug distribution within the particle, the porosity, the surface morphology, and those microstructural attributes are what determine release behavior. The FDA actually introduced the concept of Q3 equivalence for complex generic products, meaning it's not enough to have the same ingredients in the same amount, Q1, Q2. The generic must also demonstrate comparable microstructure and physical architecture. For a microsphere, that means demonstrating similarity in internal drug mapping, pore geometry, and particle size distribution. How do you characterize that at a regulatory level? You are looking at techniques like FIB-SEM, focused ion beam scanning electron microscopy, confocal Raman microscopy for 3D drug distribution mapping, synchrotron X-ray tomography. These are not routine QC methods, they are research-grade analytical tools. Alright, so let's talk about the regulatory reality. A generic drug is approved via an ANDA, an abbreviated new drug application, which relies on demonstrating bioequivalence to a reference listed drugs. The underlying assumption is pharmaceutical equivalence. The same active ingredient, same dosage form, same route of administration. For a simple tablet, this is clean. For a PLGA microsphere depot injection, no. The Definition of same dosage form becomes philosophically complicated. The FDA's own guidance distinguishes between Q1, Q2 and Q3 sameness. Q1 means qualitatively the same inactive ingredients, Q2 means quantitatively the same, and Q3 means structurally equivalent. Same microstructure and physical performance. For PLGA products, achieving all three simultaneously while also demonstrating bioequivalence has until very recently been a regulatory barrier. The landmark moment in this space came in 2023 when FDA approved first ever generic PLGA Microsphere product, a generic version of Vivitrol, a naltrexone depot. That was a watershed event. It took over a decade of FDA research under the GDU-FA program to build a scientific foundation for that approval, and naltrexone is a small molecule much simpler to encapsulate than a peptide like leuprolide. Leuprolide acetate is a 9 amino acid peptide. Its interaction with the PLGA ionic hydrophobic conformational is far more complex. The burst release behavior, the degradation kinetics, the physical stability inside the polymer matrix, all of these are peptide specific. IVIVCs developed for naltrexone microsphere cannot simply be transferred to leuprolide. The science has to be rebuilt with scratch. Let me put the bioequivalence challenge in the concrete terms. If you are developing a generic for leuprolide depot 3-month formulation, your bioequivalence study needs to demonstrate equivalent testosterone suppression over a 12-week window. That means dosing patients, collecting blood samples over 3 months, and doing pharmacokinetic modeling. But drug release from a PLGA microsphere is not linear. You have burst, the plateau, the degradation phase. A patient might show equivalent Cmax and AUC overall, but different release profile in the first 2 weeks, which could mean a different testosterone flare risk. Does that matter clinically? Probably. Does it matter regulatorily? That guidance is still being written, and the variability inherent in long-acting parenterals between subjects, between injection sites, between lot-to-lot polymer batches inflates the confidence intervals in bioequivalence studies. You need larger patient numbers to achieve adequate statistical power, more patients, longer studies, higher costs, and many more. Development timelines stretch to a decade or more just for one dose strength. Leuprolide depot comes in multiple dose strengths. For example, 1 month, 3 month, 4 month, 6 month formulations. A generic developer would need to independently characterize and run bioequivalence studies for each. The investment required is enormous, which brings us to the third category of barrier beyond science, patents, know-how, and infrastructure. So even if a company solved every scientific and regulatory challenge, that is very big, even if they would still face significant commercial and infrastructure barriers. Secondary formulation patents are real, while the original leuprolide patents have expired. Patents covering specific aspects of the depot formulation, the delivery device, the dual chamber release design, the lyophilization process, can extend to de facto market exclusivity. Then there is a technique manufacturing know-how. PLGA microsphere production at commercial scale requires specialized equipment environmental controls, and process expertise that took the innovator decades to accumulate. That knowledge isn't in any patent filing. It lives in the heads of experienced formulation scientists and in the standard operating procedures of specialized facilities. Building those capabilities from scratch is a years-long undertaking. And finally, the number of contract manufacturing organizations globally that have validated PLGA microsphere manufacturing capabilities is small. The specialized production infrastructure simply doesn't scale the way it does for tablet and capsule manufacturing. So where does this all go? Is a generic Lupron Depot just permanently off the table? Not necessarily, but it will take time, scientific investment, and regulatory evaluation. On the analytical side, we are seeing real advances. AI-assisted image analysis of FIV-SAM data is enabling faster Q3 equivalence assessment. Accelerated in vitro release methods are being developed and validated. IVIVC modeling for peptide-loaded PLGA microsphere is an active research area. These tools are paving the way for a more efficient generic development pathway. On the regulatory side, the FDA has signaled its intent to issue product-specific guidance for complex generic products, including PLGA-based injectables. The Vivitrol generic approval in 2023 demonstrated that PLGA generics are achievable, and it provided a regulatory precedent and scientific framework that future applicants can build on. The question isn't whether a generic leuprolide depot will ever exist. The question is who is patient enough, scientifically capable enough, and financially resourced enough to do it. Given the scientific complexity we have discussed today, the answer is very few companies, which in its own ironic way explains why 40 years later we are still having this conversation. On analytical sciences and you want to talk through the characterization challenges, reach out to us at Resolve Mass Laboratories. This is exactly the kind of problem we spend our days solving. And if you found this useful, drop a comment, share it with a colleague, or let me know what complex product you'd like us to tackle next. Thanks for watching. If you're navigating complex drug development challenges from IND enabling studies to NDA submissions, connect with Resolve Mass Laboratories to accelerate your program with confidence. Thank you so much for watching, and goodbye for now.